Hello, everyone. This is Craig Brelsford with Redshift. Thank you for joining today's event with Calidi Biotherapeutics, which trades on the New York Stock Exchange American under the ticker CLDI. With us today, we have Stephen Thiesing, Chief Business Officer, and Andrew Jackson, Chief Financial Officer. We will begin with a brief presentation in a moment, and then we will answer your questions. Users may submit a question at any time by using the Q&A tool at the bottom of the Zoom window. Before we begin, please allow me to read the Safe Harbor Statement. This call may contain forward-looking statements within the meaning of the Private Securities Litigation Reform Act of 1995. All statements pertaining to future financial and/or operating results, along with other statements about the future expectations, beliefs, goals, plans, or prospects expressed by management, constitute forward-looking statements. Any statements that are not historical facts should also be considered forward-looking statements. Of course, forward-looking statements involve risks and uncertainties. I now turn this webinar over to the Calidi team. Gentlemen, please go ahead.
Thank you, Craig. Thank you, Redshift team, for facilitating this webinar. And thank you, attendees. So you should be seeing systemic and localized anti-tumor virotherapies. That is what Calidi is about. We'll explain a little bit more what these things mean in the coming slides. The key here is that our technologies are designed to attack every tumor and arm the immune system to kill cancer. We've already had the safe harbor. We do have the forward-looking statements and risk factors. And now on to the overview. Calidi is a clinical stage biotech company, and we have innovative oncolytic virotherapies. We use these therapies, which are designed to attack tumors, and our cell-based technologies actually protect this oncolytic virotherapy payload so that it can reach a tumor site. And that's really historically been the challenge of OV therapies, is that they are taken out, they're rapidly eliminated by the patient's immune system. So Calidi's technologies are very differentiated and novel in that they overcome this challenge with the immune system so that our technologies, our treatments can get to the cancer. We believe that the systemic, which is an IV-based administration technology, and our localized technologies will revolutionize treatment of solid tumors and cancers in general. This is one slide that really summarizes how Calidi technologies are set apart. We overcome these obstacles to traditional oncolytic viral therapies. And for short, I'm going to call them OV therapies. So if you look on the left, the unprotected oncolytic virus, you could administer a large amount of OV, and within a short amount of time, that OV is going to be rapidly and dramatically eliminated by the immune system. Our own Dr. Boris Minev, who prior to Calidi was with Genelux Corporation, saw this firsthand, where within 1 hour of administration of an OV, 95% of it was taken out by the immune system. And the result here is that the tumor is untouched. Calidi's technology, starting with our Neuronova and Supernova platforms, has used a stem cell loaded with a small amount of oncolytic virus, and then we incubate that over time. And so we get this bonus of a much larger amount through an incubation process where that OV will amplify, it will continue to expand. So what we get here to start are two very significant advantages from using the stem cell. The first is protection from what's the human immune system, which scientists refer to as the human humoral immune system. And we get this protection acting much like a Trojan horse or a cloaking device so that the immune system is not attacking this OV payload and taking it out. So we get the protection, we get the amplification. Remember, we start with a small amount of this very expensive asset, and within a short incubation process, we get this fully loaded stem cell. And then the third element is that this stem cell actually primes, it opens up the tumor microenvironment. And so that tumor will actually embrace the stem cell, pull it in, and within 24 to 36 hours at human body temperature, that OV will continue to replicate and expand within the stem cell until it gets to the point where it blows out the walls of the stem cell, much like a water balloon, if you will. But that OV expands, blows out the walls of the stem cell, and then the OV, the red dots represented here, will begin to consume the tumor from within. And this is the key of Calidi's solution, in particular for neuronova and supernova, which I'll get into shortly, and then we'll talk about the systemic a little bit later. This is a look at our neuronova and supernova platform advantages. So looking in and across the landscape of the OV oncolytic virus players, we've been most closely compared to by our covering analysts at H.C. Wainwright, also at R.W. Baird, to Replimune at the time of going public. And the key differentiator here being our stem cell technologies. While most of the other companies have an engineered virus, we have the benefits from our platform technology from the allogeneic stem cells of reduced viral elimination by the immune system, those virally encoded proteins expressed at administration, effectively opening up the tumor microenvironment, as well as the various stem cell-derived immune modulators. As many of you folks know, Calidi went public last fall via a SPAC business combination. So we've had some downward pressure on the stock. However, we are moving forward in our clinical plans, as well as license and collaboration opportunities, and anticipate seeing upward benefit in the coming weeks and months. In terms of our drug pipeline, I mentioned neuronova, so that is CLD-101. That is technology that we exclusively license from Northwestern University and City of Hope, coming up on 3 years ago. And through largely CIRM sponsorship, California Institute of Regenerative Medicine, we are currently underway in a recurrent high-grade glioma clinical trial and have benefited from external validation and funding by CIRM to fund this trial that's currently underway at City of Hope. The neuronova in newly diagnosed high-grade glioma, that has already been in patients, a 12-patient safety study published in The Lancet, and this will go into further clinical trial in the second half of this year, going into a 1b/2. And then supernova, which is the platform technology that we built the company around, targeting advanced solid tumors, skin cancers, head and neck, triple-negative breast, and soft tissue sarcoma. This is a localized administration, and we're planned for a phase 1, a follow-on to a safety study in 26 patients that will occur later this year. We have received $3 million in CIRM validation and funding to support the final pre-IND work and IND work, and then CIRM has indicated a plan for further support as we take that to clinical trial. And then RT-NOVA is what we announced earlier this year. That's a true systemic and IV-based administration, and we'll get into more detail on that shortly. This is a look at milestones, and I'm excited to share updates on what we've already accomplished this year. And right now, looking to go into dosing of first patient in the newly diagnosed high-grade glioma. Remember, that's a follow-on to a 12-patient safety study, which will happen at Northwestern University in the second half of this year. And then I mentioned the recurrent high-grade glioma. This is a multi-dosing approach. We, of course, received CIRM funding to support that clinical trial. And we actually provided an update, a poster presentation at ASCO just a week and a half ago that was extremely well received. And we are now in the 4th cohort. So there are 4 groups of patients The first would be 1 dose, second 2 dose, third 3, and fourth 4 doses. And thus far, the fact that we're progressing into the fourth cohort shows that we have had safety recognized in the earlier cohort. So dosing the first patient in that cohort and recruiting additional 3 patients to round that out. Okay, CLD-201, SNV1, and we have completed the drug product manufacturing, getting ready for IND submission and anticipating a first patient dosing and a dose escalation toward the end of this year. From Biocom partnering meetings where we announced our RT-Nova systemic, we actually did a poster presentation. Dr. Antonio Santidrian, who's currently on vacation or would otherwise be with us today, but he was very much inundated with strong interest from a number of big pharma and other organizations. So we're excited about moving forward in licensing and collaboration opportunities for this platform. This is a look at the 3 platforms, and I'll be fairly quick on this slide. NeuroNova, which I've already told you about, this is using a neuronal stem cell, allogeneic, combined with an engineered adenovirus, which we call CRAD-SPK7. And so the stem cell is loaded with the oncolytic virus. Similarly, I call these sister technologies our supernova. Again, I should say NeuroNova is in clinical trial run right now on recurrent and going into further clinical trial on newly diagnosed shortly. And then Supernova, which we built the company around, is using a mesenchymal stem cell that's allogeneic, adipose-derived, combined with what we call our Cal1 vaccinia virus. So one of the things that you see just from a form factor, these are fairly large. And as we were briefing Big Pharma about 3 years ago, we were asked if there was any way that we could move these technologies toward a systemic and IV-based platform. And we have very much done that with RT-Nova. Again, we announced it at the Biocom Global Partnering Conference in late February. This one is a true IV-based systemic targeting lung cancer and metastatic, advanced metastatic cancers. And this is an IV-based treatment again. We have engineered an extracellular envelope vaccinia virus, and I'll tell you more about that in the coming slides. So looking at NeuroNova, again, we completed a single-dose safety study, 12 patients, almost 3 years ago. And the key here is that we saw from 1 dosing, progression-free survival went from 5 months to nearly 9, and overall from 12.7 to 18 off of 1 dose. That has led us forward in terms of a multi-dosing strategy, which we've employed at City of Hope. Again, this is in recurrent high-grade glioma. The prior safety study was in newly diagnosed. And as I mentioned, we're in the 4th cohort of treatment. So have already treated 7 and adding further patients in this 4th cohort, which will be 4 weekly doses of this treatment. What enables us to do a multi-dosing is that we have left behind a catheter in the resection cavity so that we can administer additional doses. As well, we've left behind a catheter in the ventricles to sample and monitor cerebrospinal fluid. As we move forward, we're taking the data, which was well received by the FDA, from the safety in this study that's happening at City of Hope in order to enable a multi-dosing at Northwestern University in newly diagnosed high-grade glioma, and looking to start this up very soon. And this is a look at the 4 cycles. Remember, I said 4 cohorts. So again, first cohort, 1 dose, and we're now in the 4th cohort, which is 4 treatments. So the fact that we have advanced to this 4 cohort is very much proof that we've seen safety from the dosing in the prior cohorts. And again, at City of Hope, we have enjoyed 12 million in CIRM funding to support this clinical trial. This is a look at the multi-dosing in newly diagnosed that will begin shortly at Northwestern University. At time of biopsy, we administer the first dose, and then at the time of the resection of that primary tumor, we are leaving behind a catheter so that we can, without further invasion, administer multi-doses following on. One of the things that I didn't mention is that this Neuronova technology, CLD-101, that virus is engineered with a survivin promoter so that the standard of care of radiation actually further activates and revs up that virus to target cancer. And so we're very optimistic about the results that will be seen in a multi-dosing strategy based on what we saw in the single dosing safety study. So this is a look at supernova. Again, this is a directed localized administration. And CLD-101, that, as mentioned earlier, has completed phase 1. We are doing a follow-on to a 26-patient safety study in allogeneic platform for supernova, and this will go into phase 1 in the second half of this year. As I mentioned, the safety study, single dose in autologous using patient's own stem cells, and then I'll show you the highlights of strong initial signals of efficacy from that single dose. And moving forward now to an allogeneic. So the FDA feedback was using patient's own stem cells was not a greatly scalable approach. And they wanted us to be able to scale into millions, multi-millions of doses. As well, they wanted to consistently characterize from vial to vial and dose to dose the number of cells in that treatment. And so about 5 years ago, we created a master cell bank, allogeneic, from 10 healthy adult donors under FDA guidelines. And that has been the basis for our allogeneic off-the-shelf multi-dosing approach as we move forward. So this is a look at the highlights showing tumor regression and survival, an actual durable efficacy highlighted here. And so this is a patient, one of the 26 that we treated, 70-year-old male, metastatic head and neck squamous cell. He's very late stage, 4B, so he has approximately 3 to 5 months to live. And when we dosed him at one point in time in the 3 primary lobes of this tumor, by day 45, we see some crustiness, necrosis. And by day 52, what had been a very hard tumor has now become soft and squishy, somewhat liquefied from within. And then at about 6.5 months later, we see that tumor is completely gone. And so this is a durable tumor regression and survival off of one dosing. We know from this patient keeping in contact with us that he lived for at least 3 years after this treatment. And so very impressive, durable efficacy. And so this is another patient. This is a thyroid papillary carcinoma that had spread to his lymph node above his collarbone, the supraclavicular lymph node. And this is a stage 4, also very advanced. And we dosed at one point in time. And within 3 months, that tumor has fully regressed. Another example of durable tumor regression and survival. This slide highlights the fact that the platform, Supernova, has potential in multiple indications. In that safety study, we treated these many different cancer types. Again, late stage patients, stage 3 and 4A through C. And in one dosing, we saw average known survival in the order of 24 months, approximately 2 years. And that was, even though we discontinued monitoring of patients at 1 year, these were patients who kept in touch with us. And so we see that this platform can be targeted effectively and effective in multiple cancer types. So moving forward with this platform, we are going into, again, an allogeneic approach, and we are targeting skin cancers, head and neck, triple-negative breast cancer, and soft tissue sarcoma, doing a dose escalation first in 5 indications, and then the best performing 3 from those 5 we will further target. And then in the best responding overall indication, we will take that going from a phase 1 into a phase 2. Very exciting to share with you RT-NOVA, our true systemic administration. We enjoyed multiple very positive discussions and dialogues with big pharma companies and several big pharma venture funds at ASCO and bio conferences over, well, we just finished those up a week ago. So about 10 days ago, we were in active briefings. And so we are now following up on those opportunities for potential collaboration and funding. What's key about this RT-NOVA is that it is engineered for advanced metastatic diseases or lung cancer, and it allows for a simple, cost-effective IV administration. It allows us to avoid a need for image-guided delivery. And this is possible because of our team. Our team, both here in San Diego and also outside Munich, Germany, has not only engineered, but proven the ability to manufacture this extracellular enveloped vaccinia platform technology that's expressly designed for systemic delivery. And so this is actually a virotherapy. It's an engineered tumor-selective vaccinia virus, which we refer to as RT or red tail. And I'll explain to you where the name came from in a future slide. And we chose it because of a large insertion capacity and being able to deliver multiple payloads into the tumor. And while this virotherapy has its own membrane, we have actually engineered an enveloping human cell membrane to protect that virus from the immune system. And so protection is a key element, and it also continues in the targeting to, because that human cell membrane has human surface receptors, it is able to target cancer specifically, and also, similar to our other 2 platforms, it modulates the immune system. It's also safe. It does not integrate into the human genome. And earlier this week on Monday, we issued a press release that we have a collaboration with SIGA to enable a safety switch, an oral drug that actually turns off the replication of our platform technology. And that's actually very desirable in clinical trial and to FDA. What's key about our engineered RT-NOVA red tail is that unlike a standard vaccinia virus, which spreads from cancer cell to cancer cell somewhat slowly, this red tail vaccinia virus that we have engineered produces these long tails of movement across to further distanced cancer cells. And these tails led to the naming by our science team of red tail. And it's actually named after the red tail hawks that we see very commonly here in San Diego. This is a look at the effectiveness of our manufacturing processes, which ensure that that second membrane can be engineered and manufactured effectively and be able to maximize the resistance against the humoral immune system. Here is a look at our ability to protect in a standard media. These are assays showing side by side a non-enveloped vaccinia virus in standard media combined with human serum has only a 12.4% survival rate. And in the case of our engineered red tail, the enveloped RT virus with the second membrane, we're seeing, when combined with human serum, a 79.3% survival. So very, very significant. And this shows that only these enveloped virotherapies can survive in the bloodstream against the inactivation by the human serum. This is a very critical step to achieving systemic delivery, and we have been able to do this. Looking at this next slide, systemic administration, I just want to highlight that this mouse model bearing 3 different human solid tumor types, and it is engineered to biofluoresce here and show when it is activating against and killing the tumors. And we see effectiveness against head and neck, lung, and melanoma cancers, 3 different cancer types. And I should just highlight that this was one dose that was administered systemically via the tail vein. This is a look at an immunocompetent lung tumor model that is implanted subcutaneously in this mouse model. And versus the control group, we see very significant tumor suppression occurring here, very significant versus the control, and remarkable tumor selectivity. And this is a look at immunohistochemistry. In the left, we see an untreated tumor with very little cellular activity. And then after administering the RT Nova, the ENV RT, we see the virus in yellow and a proliferation of these immune cells, CD8, CD4, and CD11. Very effective situation with our RT Nova. So with that, I want to hand it over to our CFO, Andrew Jackson, to talk a bit about financials, and then we'll do Q&A. Andrew.
Steve. So our market cap's a little over 14 million. We have about 64.2 million common shares issued and outstanding. We have no preferred stock. Our public float is around 45 million shares, and pleased that our average daily trading volume has increased to around 1.9 million shares per day recently. With that, let me turn it back over to Steve, and we can start the Q&A.
Thank you, Andrew. So for Q&A, I believe that RedChip is going to select and read some of the slides or some of the questions.
Absolutely, Stephen and Andrew, thank you very much. Yes, we have a lot of people on this event today, so we're only going to take your typed-in questions. That is, if you wish to speak to the team, go ahead and use the Q&A button at the bottom of your Zoom window. You may see it now. We already have several questions submitted. I'm going to read them aloud, gentlemen, so that everyone else can know what we're talking about, because only the person who wrote it can actually see what the question is. So I'll read it aloud, and either of you may answer it. So we've got this one here for you. Is the funding you mentioned sufficient to complete trials, or do you anticipate an offering? Also, do you anticipate an reverse split to get the price above $1?
Yeah, let me take a portion of that, and then Steve can fill in the rest. So since the March 31 financial filings, we've done 2 offerings. We raised 6.1 million in an S1, and we raised 2.1 million in a warrant inducement in just a week ago. As far as the reverse split goes, the New York Stock Exchange doesn't have the same rule as NASDAQ, where you have a dollar minimum. However, we did get a reverse stock split approved by shareholders last week, so the board has a discretion to enact that whenever we need to.
Thank you, Andrew. And to continue on that, the clinical trials that we presently have underway, the one in recurrent high-grade glioma at City of Hope, so that is supported by 12 million in CIRM funding. And our planned clinical trial in our supernova, our CLD201, for later this year, I mentioned that we saw 3 million in CIRM funding, and CIRM has indicated that we can anticipate an additional 8 million to 9 million to support that clinical trial once we have the IND approved, and that's what we're working on buttoning down in Q3. So I think that answers that, and we can probably go on to our next question.
Can you explain the downward price pressure on the stock price in recent months?
You know, it's really hard to pinpoint exactly what that is. You know, more people selling than buying. But really what we're pleased with is the response that we've received from our positive press releases recently. The Calidi hitting milestones and doing what we said we would do. So we're very pleased with the recent responses.
Thanks, Andrew. Next question.
Thank you very much. Yes. How is Calidi different from the other comparable companies you just shared in the presentation?
Thank you. I'll go ahead and take that. So the key here is in the technologies that Calidi employs to protect that oncolytic virotherapy payload, or OV payload, from being taken out by the immune system. So the other companies will use higher dosing, attempts at engineering the virus, and there's been mixed benefit to that approach. Whereas Calidi's technology very compellingly uses the stem cells in the case of the neuronova and supernova, and then that engineered human membrane, the enveloping membrane around the virotherapy payload in the case of our T-nova. And those are key differentiators that our following analysts at Wainwright and Baird point to as being significant differentiators for Calidi, that novel approach.
Thank you very much, gentlemen. Why did the healed patient die? Did the cancer spread to other areas that you don't know? And number 2, how did you implement the cure by implantation with other typical treatment like radiation and specific diet?
Okay, so some of these questions that are pointing to things that aren't necessarily things that we've disclosed. So I'm thinking, and I actually have just been joined by Dr. Boris Minev, who is our head of clinical and scientific. So in the case of the one patient that was first highlighted, that patient passed after the 3 years that we had been in contact. He was in his late 70s and passed from an unrelated illness. It was not cancer related. And Boris, anything additional? Yeah. Yeah. Well, we didn't point to that. So, what was the other part of the question, Craig?
Yes, this person wanted to know, how did you implement the cure by implantation with other typical treatments like radiation and a specific diet?
Okay, so Boris, maybe you can speak to how did we administer the treatments in supernova? How was that done? I'll actually
Yes,
switch.
the supernova treatments are administered by intratumoral injection. And also in this patient, one of the two patients that Steve presented, this treatment was followed by radiation therapy as well as checkpoint inhibitor Opdivo following our treatment. So again, the patient did not respond to radiation treatments before we applied our treatment, but after the supernova treatment, he responded very well to radiation therapy. And we know that there is significant synergistic effect of radiation with our treatments. So that is the result of resulting from this synergistic effect is the complete response for this patient.
Thank you, Boris. Okay, Craig, what's next?
Thank you very much, gentlemen. Yes, we're getting several questions about funding and things like that. So I'm going to put them all together into this one. How are you planning to fund the clinical trials, both underway and upcoming?
I'll go ahead and take that. So clinical trials for Neuronova, which at City of Hope, that is funded with monies from CIRM, California Institute of Regenerative Medicine. And the planned clinical trials, the continued trials in an allogeneic approach for supernova CLD201, those are being anticipated also being funded by CIRM. Again, we've already received $3 million in funding from CIRM for the lead-up work to the IND filing. Yeah, and also, as Boris pointed out, also to support our manufacturing efforts for both of those drugs have been supported by CIRM historically. In the case of RT-Nova, that's a ways off from clinical trial, and we are working in follow-up to pursue some big pharma license and collaboration opportunities, and we would anticipate additional funding to support those.
Thanks very much, Steve. Why is RT-Nova of particular interest to big pharma?
Great question. So back almost 3 years ago, when we were briefing big pharma on our platform technologies, they, in some ways, put a challenge out there for us and said, you know, what you folks have done is amazing. If you can move this toward a systemic approach, meaning an IV-based approach, then we consider that the holy grail of cancer therapies. And so the fact that we have extensive preclinical data around RT-Nova, and I just showed a few highlight slides for the purpose of this presentation. We actually went into more detail with our briefings with big pharma potential partners. Just extremely well received and excitement about what we've accomplished thus far with RT-Nova. So I think the fact that we have something tangible that they have pointed to several years ago as being the holy grail in cancer therapies, I think that's very positive. One particular meeting, I'll just share a quote from somebody who was pretty closed off before we shared the details of our presentation. They said, "This is very cool, and keep going. This is how innovation happens." And so that was really strong encouragement for us in what we're doing, not only in Neuronova and Supernova, but in RT-Nova as well.
Speaking of innovation, what can you share about the Calidi IP portfolio?
Yeah, I can share that that both Neuronova and Supernova have received numerous patents and even broader patent filings to expand them that are pending. But those patent families covering neuronova and supernova have been secured. And additionally, our T-nova, we did do the preliminary filings back in February to ensure that that IP is protected. And we've continued and will continue to expand the filings as we move that technology forward.
Now, what are the upcoming clinical trial readouts, gentlemen?
Well, as we shared on an earlier slide for clinical trial readouts, we did provide an update at ASCO on our Neuronova platform, on the trial that's currently underway at City of Hope. And we're anticipating, so once completion of the fourth cohort and that treatment has occurred, then there will be a follow-on time. And so we're looking to have additional data on readouts, most realistically in the first half of the new year, and possibly some additional indication toward the end of this year. But we're formally saying that it'll be early next year. I should also add that with supernova, that's just going into an anticipated first patient dosing in the follow-on, or I should say in the allogeneic approach, and we're targeting the end of this year. So readouts then will be, early indications would be toward the end of next year.
Thank you very much, Steve. Thank you very much to our participants. We've got a lot of questions here today, and we're going to try to get a few more in. We may even go 5 minutes over the hour, if that's all right, gentlemen, to try to get as many questions in as possible. So the virus is infused into the stem cells, then the stem cells containing the virus is the product that will be injected into the patient's body. Is that correct?
Boris?
Yes, that's exactly right. For the first two programs, for the Neuronova and for the Supernova, this is exactly, we are loading the virus into stem cells, and the stem cells loaded with the virus are injected in patients. For the third program, for the RT Nova, the virus is specifically engineered so that it contains second envelope, which is protecting the virus from inactivation by the patient's immune system. But there is no cell in this
no stem cell.
No, any cell. It's just virus that is engineered for protection with two envelopes.
Yeah. And again, that RT Nova is, in that case, it is IV administered into the bloodstream. Thank you, Boris.
Yeah, thank you very much, Boris. Have you had a case treated without radiation, i.e., to test the effect of your treatment alone and cancel the recurring effect of radiation?
Yes, we had several in our previous clinical trial with allogeneic cells loaded with the oncolytic vaccinia virus. Several patients were treated without radiation, and especially one of them, I believe, the thyroid cancer patient that
we showed,
Steve presented, was treated with our treatment only, no radiation, no chemotherapy.
Perfect. Thank you, Boris. Okay, Craig.
And what is the purpose of the radiation therapy after cell therapy treatment? Can we just rely on cell therapy without chemotherapy?
So I was not sure because initially person was asking radiation and then chemotherapy. So really depends on the type of tumor, but we really try to apply our treatment as early as possible before radiation and chemotherapy, because as it is well known, the radiation and chemotherapy are suppressing the immune system of the patients, and our treatment is immunotherapy to activate the immune system. But we have found in many studies that radiation, especially at lower dose, is actually helping our treatment. So we really depends on the type of tumor and clinical trial design. We can combine our treatment with radiation therapy to really achieve better response.
In certain cases. Yeah. Thank you, Boris.
Final question here, gentlemen. Did you shrink all tumors in the trial?
Okay, so we represented a neuronova trial and a supernova trial. So, Boris.
Yeah, so again, there are different platforms. For the neuronova trial for brain tumors, there is actually no tumor. Tumor has been removed by surgery, and after removing the tumor, the product is injected into the walls of the surgical cavity. In this case, we are targeting small groups of tumor cells that are remaining after surgery in order to prevent recurrence of the brain tumor. So in this case, several patients who were actually of a group of patients with unmethylated MGMT promoter, meaning they are not responding to chemotherapy, their life expectancy was increased, and also progression-free survival and overall survival. So this is a very positive sign. For our other program, many of the patients who were treated with intratumoral, with supernova, had tumor shrinkage, but not all the patients had extensive tumor shrinkage, which is expected. But I have to mention that in this clinical trial, we used very, very low dose as a first clinical trial. And also we used patients' own stem cells, so we couldn't really control the number of viral particles that are inside each one of these stem cells. Now it's much better characterized product, and we have about 10-fold more viral particles in each cell than before in the previous trial.
Super powerful advancement. Thank you, Boris. Okay, with that, we're at 2:04. I just want to present the last slide here. And that is, Calidi is very much driving a clinical revolution in cancer treatments. And this is because of our systemic, that IV-based RT-Nova platform, as well as our localized, our neuronova and supernova platforms. All of these delivering a breakthrough in oncolytic virotherapies. And it really means that our technology, and as Dr. Menev pointed out, Boris, depending on the cancer type, depending on the tumor, and depending on the clinical trial regimen, we will target these different therapies to different patients. And it's very much about engineering what's most effective. So again, designing these platforms that attack every tumor and arming the immune system to kill cancer, that is what Calidi is about. And just want to thank you all for your time today.
Thank you.
Bye-bye.
Thank you very much, gentlemen. And before we wrap up, let me just give the audience here a few ways to learn more about Calidi. You can call us at 1-800-REDSHIP. That's 1-800-733-2447, or email us at CLDI@redchip.com. Please visit RedChip's investor information page for Calidi Biotherapeutics. It's CLDIinfo.com. There you can view and download the investor presentation and fact sheet. as well as sign up for news alerts. Thank you again to our many participants today, and thank you very much, Calidi team.
Thank
Thank
you,
you.
Craig, and thank you, RedChip. Goodbye.